Hi, my name is Sylvie Le Guyeder, and I am the manager of the live cell imaging facility at the Karolinska Institute in Sweden. I'm going to show you how to, or give you a little introduction about how to analyze an image using the NIS-Elements software, which is the Nikon software for microscopy. Here we have an image, and it has 4 channels. The nuclei in this tissue are in blue, and then we have a protein that is labeled in green, and there's a high background. And then here we have in red another protein that labels the astrocytes, and this is very dim. So the first thing I'm going to do is I'm going to push the brightness so that I can see something in there. And to do this, I'm going to use the lookup tables, so I can right click in the background of the software, and I go to visualization control, LUTs for lookup tables, and I get this guy here. Right, so this is the red channel, and I'm just going to push that. So that doesn't change the data, it changes the display. It makes it easier for me to see what the label is. And then I have a 4th channel, which is far red, labels yet another protein. Okay, so I can do the same thing here. Okay, so now I can show all of them. That's what it looks like. And what we would like to do today is that we would like to have a look at the intensity of the green channel and the intensity of the far red channel, but only in the astrocytes. So to analyze this data, what we're going to do first, and then we have a lot of images that we will analyze in the same way. So to analyze this data, we are going to open something called general analysis. So if we go under image, we can see here we have 2 different things. One of them is the general analysis, the other one analysis explorer. While once we have some analysis, general analysis saved, then we can use the analysis explorer to go back there. So let's start with going to the general analysis. This comes up. Okay, so we have, as I said, 4 channels, and the software recognizes this automatically, so we can create different channels, analysis channels. So if we click on this button here, we see that we have the possibility to analyze each of our channels. So right now what I want to do is that I want to find the astrocytes so that I can interrogate the intensity of the green and the far red inside the astrocytes. So the astrocytes are in red, so I'm going to select TRITC, and then I say OK. So now I have my first channel here. Every time we create an analysis channel, we are getting this display here. Okay, so I can, for example, I could now select a 2nd channel if I want to analyze the blue, the nuclei. Then I select here, I get a 2nd one. See, it looks the same. Okay, so let's delete this one for now. I have this analysis channel, and in each channel we have always 4 areas. This is the area number 1. Here we have a 2nd area. Here we have another one, and here yet another one. Okay, so we're going to go through all these areas. So the area that is, let's say, crucial is the 2nd one here. This is the area that will allow us to do the segmentation of our channel. So if I go here to TRITC channel, what I'm going to try and do is create a mask on this image, where a binary mask, which means an image that where I have separated for each pixel, I am given like a value of either 0 or 1. So I'm going to have 1 to associate 1, which is pixel, when I say this is my signal, and then 0 when I say this is not my signal. Okay, so the idea of the whole area here, the segmentation area, is actually to create this binary mask and decide what is the signal that we're interested in, what is not the signal we're interested in. Okay, there are several methods to run the segmentation and make this mask. One of them is called thresholding, but if you click here, you have different ways to select different things. Okay, and in here we also have other ways to select what it is. The simplest method for segmentation is thresholding, and all it does is decide above this intensity, this is my signal, below this intensity, it's not my signal. So it's a very simple way to make a mask. And you see that if I slide this now, on the image, you can see that I'm selecting more and more of the pixels. So basically, I am saying now that everything that is above 221 as intensity is considered as signal. And everything that is below is considered as background. So this very simple segmentation method called thresholding. And you can see the mask here. So my mask is over all the pixels that have this intensity above 200 or here 167. Okay, we're making here a biased decision. This is something very important to understand, is the segmentation always has the input of the user. But you're making the bias decision over the whole image. So it is still a good method. Okay, so now we have something. If I want to actually see my signal underneath, I can tick and untick this mask by doing this. And I can see it's not that bad. So all these very dim things here, we have not selected that. So I could, of course, increase even more and select even more things. So now I'd get even the dim ones. But maybe I'm not interested in the dim ones. Maybe I'm only interested in the bright ones. So I can do something like this. And you have to remember that I have pushed the display. And depending on what you show, it will look like you have selected everything or not. So again, this is something that you have to consider, that we have a bias there. Okay, so now I say, this is not bad. I like it. So I have done my segmentation here. The area before this, which is called preprocessing, is an area where we're going to do something to the image to help the thresholding. Okay. Once we have managed to do some thresholding, we can then analyze this binary image, which is the red image, which only contains one pixels that are red and that are considered as signal, and zeros, pixels that are black and considered as background. Okay, so we are going to analyze this binary image, and we're going to decide, I'm going to throw away all the small things, for example. So if I click in here, I can say filter objects based on the size, for example, the fill area. Okay, and now if I click in here, I have all this the histogram of all the objects, the size that they have, how many objects have each size. And you can see there's one of them here that is much bigger, and it's this guy here. So I can now do this, and you see that it's deselected. So I can filter my binary objects in the binary image by using all these filters that we have here. So I have a lot of different types of filters, tons of them. Okay, so you can play with them and figure out how you want. For example, you can select only the objects that are very rounded or very elongated or something like this. There's lots of always a special orientation. Okay, so now I have thrown away the very big stuff. I'm happy with that. The last area in here is, what is it that I want to interrogate? So basically, now I have managed to identify where my astrocytes are. What I want to know is, what is the intensity of the green there, and what is the intensity of the far red? So what I can do is I can go to this area, and then I go here, and I say, for each object, I want the intensity, and I want the mean intensity of the green, and I also want the mean intensity of the far red. And now I have the mean intensity and the of the green and the far red over the whole field per small objects, which is per small red dot, let's say. So I'm happy with that. And now what I can do is I want to show you how I could also analyze the nuclei so that you get an idea of how we can use also this area. Okay, so I'm going to now look at the nuclei, and for this I'm going to analyze the blue channel. So I add another channel based on the DAPI. Okay, now the red is a bit disturbing here, so I don't want to see the red right now. So what I'm going to do is that I'm unticking this box. Okay, so the red disappears. Now you have to be really careful because there are two different things here. One of them is selecting the channel that you want to modify, and all these here relate to this channel. Okay, and this is done by having this line white. Okay, it's not the same thing as displaying the mask or not. This is done by ticking and unticking the box. Okay, so we have two different things. Make sure that you have the correct channel settings showing here, because it's very easy to modify the wrong channel by mistake. Okay, so right now I don't want to see the red and I want to modify the blue one. So I could now do the same thing I did before. I'm going to use the very simple method, a bit of thresholding. I push this a little bit like this. Okay, right. That's a little bit of a problem, because you see, if I push too much, then I get all sorts of things that obviously are not nuclei. If I don't push enough, I get weird stuff like this. And this is because nuclei typically are not even. So they have nucleoli, and the DAPI is not even inside the nuclei. So what I would like to do, if you squint a little bit and you look at this image, you can see very well where the nuclei are, and you cannot see that the label is uneven. So that's what we want to do. We want to force the software to squint. And what we're going to do is that we're going to use this area that will help us run the segmentation properly. So pre-processing is something we do to the image before we make the mask, and this is meant to help make the mask. Okay, so we're going to do a little bit of pre-processing here. As you can see, again, we have lots of different things that we can do in the pre-processing, like removing the background, increasing the contrast, etc., etc. There's lots of stuff, but right now one thing that works quite well for nuclei is under denoising median. The median, if you look now at the image as I click, you will see that it becomes a bit blurry, but the edges are very well preserved. So median is really good for nuclei because it makes the inside of the nucleus blurry, so more even, more homogeneous, but it preserves the edges. So I can increase this filter and make, if you look at the image, you see I make things even more blurry in the inside. So I homogenize even more, okay? And I can, by clicking here, I can toggle between the original and not the original. So now I'm going to use this to make my segmentation. So if I show my mask now, you see that the edges are a lot smoother and I don't have all these small dots everywhere, right? So it's much easier now to segment the nuclei. I still have some bits that go a little bit wrong, like tiny things, but this is a tissue, so tissues are difficult in general to segment. I also have stuff that are have a hole like this, but I can fix this afterwards. Okay, so now that we have done our segmentation, something like this, okay, I can so my pre-processing helps the segmentation, and just with the simple threshold method, now I can segment. And if I want to fill in these holes, I can just include this fill hole. And you see everything that has a hole and is surrounded by, completely surrounded by the mask will be filled. So now we have done some pre-processing, we have done some thresholding. We could, for example, filter out all the small bits that we don't want, okay, or the long ones or the very big ones. So if I go here and again I say filter objects, I can filter based on the size, okay, and then I say fill area and I get my histogram. I'm going to remove the big one. Yeah, that is maybe not what I want. And I also maybe remove the tiny ones. Yeah, so all the small ones go away. Okay, let's say that we're happy with that. So we've done now some processing of our binary. We've filtered out what we didn't want. And now we're going to decide what we're going to save. So maybe, for example, we could decide that we want to count the nuclei. Okay. There is improvement here. Some are improvement possible. Some of them are fused or not. And again, you can toggle on and off to figure out what you want to, if it's correct or not. Even though we've done some pre-processing, I want to mention that the final image that is interrogated is the one without the processing. So this is only here to help run the segmentation. Okay, so we have now these two guys. What could we do with that? For example, we could, let's say that we are only interested in interrogating the GFP and far red intensity in these astrocytes that have a nucleus. Okay, so we're going to do a little trick for that. I'm going to duplicate this channel. So if I click here and I select again the treats, I now get the possibility to copy these settings. Okay, so I'm going to tick this in and I'm going to call this expanded. I will show you why. So now I have these two channels that are based on the red channel, and one of them is called treats and shows me where the astrocytes are, and this one is exactly the same right now, but I'm going to modify it. So first I don't want to show these two because they are also in the way. And I go back to my red channel, and what I'm going to do is that I'm going to make these cells a bit bigger. And that is because I want to see where the nuclei are. And you see the nuclei, very often they are not under the red mask. So I would like to find the cells that have a nucleus. And for this I'm going to increase the mask and make it a bit bigger, like a few pixels bigger, so that the mask expands over the nucleus. And then I'm going to only get the astrocytes that have a nucleus. So here we go. Everything is the same, so I keep the way it is here. But in here, I'm going to add a basic dilate function, which allows me to grow my mask. I just go back to the red. So I grow my mask by a little bit. You see, if I toggle it on and off, you can see this is the mask that I have that shows me where the red label is. And here, each little part of the mask has been expanded by 1 pixel. You can see this is the pixel size. So by default, if you do like this, you're expanding by 2 pixels, 3 pixels, but you can enter anything here. So now I'm expanding by 2 pixels. Okay, so my mask is much bigger. Now what I can do, so you see the mask is now starting to cover areas where we have nuclei. So I'm quite happy with that mask. I don't really need to save this again, so I'm going to delete them. All I want is to find where the nuclei are. Okay, so I don't need to save anything. I just want to get this mask. Okay, so now I have my mask, which is expanded, and then I have the nuclei. And what I can do is that I can combine them. So I can say, okay, I would like to have these, to get only the astrocytes that have a nucleus. So I can go here, and I can do a combine channel. So this comes up here, and now I'm going to say, I want to get my expanded mask that has a nucleus. Okay, and if I untick the other masks, I can see that now I only get the astrocytes that have a nucleus and not those that do not have a nucleus. Okay, I show you the difference between the expanded mask. We have a lot of different things that show up and they don't have a nucleus next to them, and then the mask with the astrocytes that have a nuclei. So now that I have this, I could decide that I'm going to have the object intensity, mean intensity of GFP, and then I have the object intensity, mean intensity of the far red. So what I have done now, if I want to go through the flow again, is that I have identified first the astrocytes, okay, and then I have identified the nuclei. Afterwards, I have expanded the astrocytes so that I cover the nuclei, and then I have filtered out all the astrocytes that do not have a nucleus. Okay, here we go. What I'm missing here is that my astrocytes are expanded, so I would like to shrink them again, and I can do that here. And I go erode, and I go down. Yes, now I found my astrocyte that have a nucleus, but they are the original size. Okay, and now I save under this new mask, I save the mean intensity of GFP and the mean intensity of the far red. So I have managed to identify the astrocytes that have a nucleus, and then I can look at the far red and the green. Okay, so now what I can do is that I can save this. So if I say save, it asks me for a name. Somehow in NIS, this goes into a big bag and everybody will get all their analysis together. So I prefer to also export and put it in my folder. So I can just say OK here. And now I'm going to export. And I will save this as Patricia astrocyte green and red. So I've already done it before. So I'm just going to override this. I say save. Ask me, do you want to override? Yes. Okay, fine. So I have now exported this into my folder, and then I can use this button to import. This is because I really want to make sure that I have my analysis saved in my data folder. Okay, so now all I need to do is press run now, and then the analysis runs. So now we see all the masks together in here. And then we have here, when we have a segmentation done, we have several buttons. One of them that shows the overlay between the image and the masks. Here we have only the image, and even though some mask is left. And here we have the mask, okay? In here we have this little button with binary layers. We can now decide what we're going to display. So what we're interested in is only the combined. So I only select the combined, and that's what I'm seeing. Okay, so now when we run the analysis, this window popped up. It's called Automated Measurement Results, and here we have all the objects that we have analyzed in this channel, combined analysis channel. Okay, you can see this here under object data. There are different ways to display all these results. So object data will show you the data for the mean intensity of the far red, mean intensity of the green for each of these objects. You can interact with the image. If you double click in these lines, you can see at the top here that the objects are showing. So the object that is identified, okay? You can also click in the image and it jumps to the line. So you can easily pick an object and see what the results are for this object. That's very useful. This is what you see under the object data, and otherwise you can aggregate all these because it's a bit difficult to see, to make sense out of it. So you can aggregate all this using object statistics. And now we have only one image. So this is for all the objects, the mean intensity of the far red and of the green. And otherwise we can see things on the whole field. So what it shows us here is simply the proportion of the area that is covered by the red mask. Okay. Okay, so that's what we have. Now we're very happy with all this, but we've lost our analysis window. If we want to find it again, we can go back to image. Now we go to the Analysis Explorer. Every time we run an analysis, we're going to get a line like this, okay? And this is the analysis that we ran. So if I double click on these lines, what I'm getting is this. If I am double clicking on this line, what I'm getting is the analysis itself. So I can run it again, modify it, and etc. if I want, okay? So that's the way to go back to this window, is simply to go back to the Analysis Explorer. You can import it from your file if you have saved it, and then you can run it again. So you see, this is what I would do, for example. For this one, I say import, and then I go to my folder, which is on the desktop. And Patricia, hoop, and there we go. Alright, so I haven't run that one. I would have to run it again if I wanted, but this is the analysis that I have saved. Okay, so now that my analysis is saved, I can batch this. Means that I can apply this very same analysis to lots of images automatically. And that means that then you're not biased. You're applying everything in the same way. So how am I going to do that? I simply open, so I'm going to close that guy. Show you, I close everything here. I forgot to say that in here, once you have the data, you can of course export them using this export data to Excel, and here shows. And then we have, so I think it's hidden behind. Yeah, here we get all the data that we want. Okay, so now I go back to my stuff, I close this, I have my analysis here that I have loaded, and now I get this button called batch. So what I do is that I open this and I'm pointing to the folder, let's say in here, where I have several images, and it recognizes automatically which of the images can be analyzed, because these have 4 channels, the others have only 3 channels, and here I need the 4 channels. So it recognizes all this, and then I say, for example, I can filter based on the format as well, and I can have a prefix and all sorts of different filters to find the correct images in the folder, and then I just say run. Okay, and then it is processing each of the images one by one. Right, and now we get the result in here. I say close, and now if I look at the statistics, I now have an analysis aggregated per image. Okay, and that I can export again. So this is the way to use the general analysis and the to analyze images in NIS. There are a lot of different filters that you can use, and there's a lot of different things that you can do. It's very important that you start analyzing your data while you're doing your imaging. Do not acquire lots of images first and then try to analyze, because the analysis will depend a lot on the way you have imaged. And so it's extremely important that you define your scientific question very well, and that you evolve the analysis together with the imaging. You will have to go back to the microscope to re-image because you realize that the analysis is not good enough, etc. So there's a constant dialogue between the way you prepare a sample, your scientific question, the way you image, and the way you analyze. Don't separate these. They belong together, and they have to be worked on all together. Okay, so I hope that you get a little bit of a better idea now of how to analyze images with NIS, and I wish you good luck.